The Human-Computer Interaction Lab at the University of Maryland has been developing an interactive visualization tool for hierarchical clustering results. This year, we released a new version of Hierarchical Clustering Explorer, HCE 2.0. This brief demonstration shows you how HCE can help you find interesting patterns in your dataset. Hierarchical agglomerative clustering was first suggested in 1967. It is an intuitive merging scheme. Let's assume we have 5 points in a plane, and we want to group them naturally. At first, we merge the closest 2 points, A and D, and make them into a new cluster. Please notice that the small height of the branch indicates the closeness of the 2 points. Next, we merge this new cluster and C, since they are the closest pair at this moment. Next, we merge B and E. Finally, we merge these two groups to have one big final cluster. One of the advantages of the hierarchical clustering is that you can interpret the clustering result in many different ways by looking at this tree from various heights. You can have one big loose cluster, or you can have two small tight clusters. Hierarchical clustering has been successfully applied to many research areas, including data mining and image processing. It has become the de facto standard of gene-chip data clustering since Dr. Eisen first applied it to gene study. There are many software tools to support hierarchical clustering, but most of these tools show only static images of the clustering results. Users must use their fingers or pencils to investigate the clustering results. Hierarchical Clustering Explorer is an interactive visualization tool for hierarchical clustering results. Here is a clustering result of 77 cereals. Let's find groups of cereals that contribute to our healthy colon. Among these available 7 nutrition factors, we choose dietary fiber and potassium and run the hierarchical clustering algorithm. Each column is a cereal, and each row is nutrition factor. Bright red indicates high value and bright green indicates low value. By dragging down this minimum similarity bar, you have tighter clusters. Right now, you have 3 pretty tight clusters. You can see that these 3 cereals in the third cluster are the most desirable ones, with much dietary fiber and potassium. They are 100% bran, and all bran with extra fiber. Interestingly, they have high protein and low calories. The second cluster has cereals with relatively high dietary fiber and potassium. And the first one has the least healthy cereals. In HCE, you can generate scatter plots and see the distribution of your data from various projections. Here, we have 2 scatter plots together with the hierarchical clustering result. X-axis of this scatter plot is potassium, and Y-axis is dietary fiber. X-axis of the second scatter plot is calories, and Y-axis is sugars. By clicking on the third cluster, you can see the 3 desirable cereals are highlighted with black triangles in these 3 views at the same time. You can confirm that these 3 cereals are high in potassium and dietary fiber from the first scatter plot. Interestingly, you can also see that they are low in calories and sugars from the second scatter plot. The Hierarchical Clustering Explorer was originally developed for the visualization of gene expression profile data. This is a clustering result of gene expression profile data of mouse muscle samples. We have 12,488 genes and 27 mouse muscle samples. Each sample was taken at 27 different time points. Initially, HCE always shows the overview of the entire clustering results. You can drag down this minimum similarity bar until you get a natural grouping of genes. In addition to the scatter plot views shown earlier, HC has another view called Profile Search. If you click on a cluster, you can see the time-varying patterns of the genes in the cluster in this view. This view is especially useful for time series data like this dataset. You can zoom in a cluster by right-clicking on it and choose Show Only This. You can zoom in further by adjusting this double-sided slider bar. Again, use the minimum similarity bar to get more delicate cluster separations. Multidimensional and multivariate data exploration is difficult, but we hope that the interactive tools of HC 2.0 will help researchers identify patterns, clusters, and outliers. Please let us know if it worked for you.